Bacteria are no exception, with molecular machines involved in translation being one of the most common targets for antibiotics. Now, for the first time, scientists led by Julia Mahamid's group at EMBL Heidelberg have visualized at atomic detail how antibiotics affect the process of protein production inside bacterial cells. This study, published in Nature, also marks the first time when scientists have observed atomic-level structural changes in the active translation machinery directly inside a cell, rather than using isolated molecules in a test tube. Importantly, this approach allowed them to identify mechanisms that such machines use to talk to each other inside cells. The study was carried out in collaboration with researchers from the Max Planck Institute for Biophysical Chemistry, Göttingen, the Wellcome Center for Cell Biology, University of Edinburgh, and Technische Universität Berlin. The research also involved contributions from the Zimmermann-Kogativa and Bork groups at EMBL Heidelberg, who helped the researchers carry out bioinformatics analyses to observe what diversity in ribosomal proteins looks like across 4,000 representative bacteria. This tiny bacterium, which causes atypical pneumonia in humans, has a fully functional protein synthesis machinery, despite being only about 10,000th of a millimeter in size. Cryo-ET allows researchers to take serial images of flash-frozen biological samples using an electron microscope and combine the resulting images to form a three-dimensional view of a cell, somewhat like a mini MRI machine. With large-scale cryo-ET data from pristinely preserved cells, it is possible to capture high-resolution snapshots of different states of a molecular machine in action and combine them into a movie, said Mahamid. The Mahamid group's approach allowed them to not only spot and count the ribosomes inside the bacteria, but also see their structure at atomic resolution. By studying a large number of ribosomes, frozen, at different stages of their activity cycle, the scientists could decipher how the ribosome structure changes as it proceeds through the protein synthesis process. Inside living cells, ribosomes function as highly interconnected systems, rather than individual molecular machines, said Shui. We revealed new features in ribosomes and the diverse translation reaction paths in cells. Antibiotics in action crucially. Using cryo-ET, the researchers could observe what happens when antibiotics enter the cell and bind to ribosomes. For example, they could confirm that the two broad-spectrum antibiotics chloramphenicol and spectinomycin bind to different sites on the ribosome and disrupt different steps of the protein synthesis process. This had been predicted by studies on isolated ribosomes, but never before been observed in action inside an actual bacterial cell. It was very exciting when we were first able to see the drug molecule binding to a ribosome inside the cell, said Shui. But it was even more exciting when we found that the ribosome populations in antibiotic-treated cells are fundamentally reshaped, functionally, structurally, and spatially. The researchers observed that the interactions between ribosomes and other complexes in the cell changed in response to the. On the one hand, this can help understand off-target effects of antibiotics, and may also help design combinations of antibiotics to increase their efficiency, said Mahamid. For example, in our group we study the interaction between viruses and their human cell host, the organization of human pluripotent stem cells and the functioning of their ribosomes, and even large multicellular 3D organoids that are collaborators and we grow from cells taken directly from cancer P. Seeing antibiotics in action inside a pathogenic bacterium. Science Daily. Science Daily, 29 September 2022. ift.tt/zdm7ers. European Molecular Biology Laboratory. 2022, September 29. Seeing antibiotics in action inside a pathogenic bacterium. Science Daily. Retrieved September 29, 2022 from ift.tt/zdm7ers European Molecular Biology Laboratory. Seeing antibiotics in action inside a pathogenic bacterium. Science Daily. ift.tt/zdm7ers. Accessed September 29, 2022. Thanks for watching. Please subscribe my channel for more videos.